Hey guys, welcome to AP Biology or College Biology. We're going to be focusing in on Unit 4, AP Biology's Cell Communication Cell Cycle. Please note we're focusing in on Section 4.7, which is all about the regulation of the cell cycle. Before we get started, I want to note two things. One, this is Unit 4, the latter end of Unit 4, so we're focusing in on cell cycling. If anything in this video is confusing because you haven't really understood Unit 4 or even earlier units 1, 2, and 3, make sure to review our podcast slash playlist section where we have complete videos related to Unit 1 through Unit 4. And make sure to hit that subscribe button because we're also going to have later videos popping up for later units. And the second thing is please make sure to note the disclaimer listed below as well. And let's get started. So here's the thing. For Unit 4, we're talking about ways that we can make sure that the cell cycle runs properly. If you remember, when we talked about Unit 4 in cell cycling, we were talking about interphase. And once again, if you haven't seen that video, make sure to focus in on section 4.6 as well. That will give you more details about what we're talking about here. But we talked about the cell cycle and how it contains G1, S, and G2, which is all about interphase, and mitosis, which is the smallest portion of the cell cycle. Now we're going to talk about the cross and guard checkpoints. That's what I like to call them, where essentially we're ensuring that the steps occur properly so we don't have any mutations or we don't have any problems that occur with the process of the cell cycle or any sort of discrepancies, essentially. So if you remember, the first portion of interphase includes G1. And what happens at the end of G1 is, if you remember, we have this checkpoint. And that checkpoint is to ensure that during G1, if you remember in interphase, we're always talking about growth. Remember, in Unit 2, we talked about cells having different organelles. If you want more specifics about that, make sure to check out Section 4.6 video and even Unit 2 to understand the parts of the cell and or what really we're talking about when we're talking about that growth phase. In essence, we're ensuring that the cell will grow properly, that all the different organelles inside are there, and that we have proper nutrients and proteins that are needed for the cell to continue to grow. However, if that doesn't occur, what happens is the G1 checkpoint will say, hey, you need to enter what's called G0 checkpoint. And that essentially is the resting state. If a cell is not ready to continue on with the cell cycle, it will remain in the G0 checkpoint indefinitely until it's ready. So, for example, a cell might not have all the nutrients or organelles or just might be small in general, so it needs to wait in that G0 phase. If it's ready to continue, what essentially happens after G1 checkpoint, we have what's called, if you remember in interphase, the S phase, okay? And during the S phase, okay, we have what's called DNA synthesis, which means that DNA is starting to grow. And if you remember, in a cell, we have a nucleus where DNA replicates. To find out more details about DNA replication, that will be in a later video as this relates to Unit 5. So if you have any questions about DNA replication or anything like that, make sure to hit that subscribe button because we're probably going to be creating videos related to DNA synthesis itself, which is a whole process. It's more in Unit 5. But just for now, you just need to understand here is the cell, here is the nucleus. The nucleus is where you have DNA, and DNA replication takes place there. It's a complicated process in and of itself. And this occurs in the S phase meaning synthesis. Towards the end of the S phase, beginning of the G2 phase, we have what's called the G2 checkpoint. And this makes sure that everything is ready to go. Now, it's towards the end point, beginning of G2, around G2. It really fluctuates, but just remember it's around G2. And the portion of the G2 checkpoint is to ensure that everything has occurred in the S point that needs to occur. So the DNA has replicated properly, there's no mutation, there's no improperness, and it's ready to go through the M phase, mitosis. And if you remember, mitosis is prophase, metaphase, anaphase, telophase. If you don't remember this, please make sure to check out section 4.6, which covers this video related to the steps in mitosis. But for now, you just need to know that metaphase is a portion of mitosis. And mitosis has prophase, metaphase, anaphase, telophase. During that second step of metaphase, if you remember, we have a chromosome lining up portion. And during the metaphase checkpoint, what essentially happens is we're wanting to make sure that the spindle fibers are attaching to those chromosomes properly. That's what the metaphase checkpoint essentially is. So these are the cell cycle checkpoints that you'll need to remember. Now, let's talk about something that might be a little bit more complicated. It's really helpful to look at this picture on the right here. I'm going to try to explain the relationship between CDK and cyclin, which are always in the cytoplasm, and these are sort of like things that come about or rise about during the different phases. They help alongside checkpoints to ensure that the phases properly occur. And it's sort of confusing because you have all these letters. So just for the purposes of this video, I'm going to start off with the S phase. Okay, because it starts off with cyclin A and CDK2. Cyclin A goes with CDK2 during the S phase. So you need to remember that cyclin A connects with CDK2 during SCDK to ensure and activate and see that DNA replication, which occurs in the S phase, occurs properly. Okay. Now, if you remember, after the S phase, we have G2 phase. Now, cyclin A still remains, but we now switch CDK2 to CDK1 to prepare for mitosis, including that DNA repair, right? So it's the same steps as before, but we're just talking about its associated cyclins and CDKs. Okay, so G2 is going to have that cyclin A still, but CDK1. During mitosis, you have what's called cyclin B that now comes in, no longer cyclin A. And during that portion, we have it still attached to CDK1. So if you notice here, the trend is we either swap out a cyclin or we swap out a CDK in every step, or most of the steps, I should say. So like, for example, between S and G2, we so far swapped out a CDK1 for CDK2 to CDK1, and then G2 to M, we've swapped out a cyclin A to cyclin B, but still kept the CDK1. I know that's confusing, but that's just a way to help you out to remember the transition between the two. Now what's going to happen is after we have the cyclin B attaching to the CDK1, what's happening in that M phase is we're talking more about prophase. And remember, we have prophase, metaphase, anaphase, telophase. Those are the and cytokinesis, which are the 4 to 5 steps involved in mitosis. We're talking about that very first step. We're talking about the prophase portion. Because if you remember, prophase is when essentially you have the nuclear envelope that's going to disintegrate and spindle fiber formation. So that's the relationship with cyclin B, CDK1. During the second transition, now remember, we have prophase, now we have aligned to metaphase, and we're talking about the transition between metaphase and anaphase. So what I mean by that is if you remember in the cell cycle, and once again, make sure to see video 4-6 for more details. It's in our playlist related to Unit 4. Now, if we're looking for, just as a brief reminder, we have metaphase, and then in anaphase, we have something like this, where in metaphase, we have the chromosomes line up at the metaphase plate. They're getting ready to attach to the spindle fibers, or they are attaching to the spindle fibers during metaphase. But then during anaphase, what's happening is those chromosomes will end up splitting apart due to proper spindle fiber allocation. And then what happens is the chromatid will separate, and then eventually what will happen is during anaphase, eventually into telophase, you will have one cell turn into two. So the metaphase into anaphase transition basically has cyclin B by inactivating the CDK. So cyclin B in the APC/C inactivates the mCDK, and that's what allows for the separation. I know that seems complicated, but what you need to know is that cyclin B is now working to ensure that metaphase into anaphase is occurring properly and encouraging the chromosomes to separate properly. Okay, so now we go back to the G1 phase. And here, during this portion, what's happening is you have cyclin B going into cyclin C and CDK3. Okay. And during this portion of the cycle, it's now going to shift. So this is where it gets complicated because G1 is a very long portion where you have now cyclin D attaching to CDK4 and 6 before transitioning back into the G1 into S phase where now you have cyclin E. So now we're switching out the cyclins from C to D to E, and we're also transitioning our CDKs. So this is where it's not always the case that you're swapping one towards the end or towards the beginning, depending on how you look at it. You're actually swapping out both the cyclin and the CDKs. So you have CDK3, CDK4/6, and CDK2 happening between the end of transition between mitosis, G1, and S phase. So it gets a bit complicated there. So to help understand it a little bit more, we're going to take a look at this graph here, okay? So it helps to understand where in the phases where the concentration of just the cyclins are very high. So for example here, we have this idea that cyclin A is going to be the highest during the S phase. That's why we started out in that S phase where we have we have that transition where it's peaking with cyclin into that S phase, and then once it's G2, it's sort of dipping essentially. So we have the G2 phase, and there's a sharp transition where now what's happening is the activity in G2 and M. It's sort of like transitioning where cyclin B is going to like peak simultaneously in G2 and then sort of pivot throughout the duration of mitosis. Then if you see here, cyclin D, for example, is going to be peaked at G1 and then tapering off, right? And then gradually decreasing. And then cyclin E is peaked during that G1 to S transition. So it is kind of confusing where they peak. So I will say you can look at the graphs to see where they peak or where they transition and where they fall associated with each of the phases to help you out because it's sort of like a transition between the two. Okay, now that we've gotten over the complicated stuff, let's talk about genes and proteins, which are key factors in relationships to cell division. The first major one is p53 protein. Just as a disclaimer, you'll probably see p53 protein when they're talking about cancer problems. Specifically, if you get an FRQ and you see p53, you should immediately think of malfunctioning p53s, because if you do, that means that you are losing your checkpoints or something is wrong with the checkpoints, which means that you have damaged DNA, which could increase the risk of cancer. So I want you to think of that. When we also talk about p53, we talk about apoptosis, which means programmed cell death. I know it seems like a negative term, programmed cell death, but we want cells to die because the thing is we don't want defective cells to continue to replicate. We want them to die to give cells an opportunity to repair and create new cells. This will allow for proper cell division and DNA damage to be regulated and then halting cell division and repair. So that's the p53 protein. We also have genes that regulate cell division. We have unlimited growth, which is growth promoter genes, tumor suppressor genes, like p53 we just talked about. If it's off, it ignored checkpoint signals, apoptosis genes, which we need. If off, then they undergo failure to undergo apoptosis exists and it could lead to things like cancer. We have chromosome maintenance genes like things like telomerase, which basically leads to unlimited divisions, and touch sensor genes that, if they're off, they have the inability to limit cell density. So these are some key gene regulating cell divisions. Now, if you want to talk about cancer and apoptosis more in detail, cancer in general is when you don't have the programmed cell death and you have these uncontrolled cell divisions that lead to cancer, metastasis, and you also have apoptosis, which is the opposite, which you want to have, which is that programmed cell death. So it's ensuring that cells are balanced in multicellular organisms. We want that programmed cell death because we don't want the continued division of cells to the point that there is cancer there. We want apoptosis because it allows for proper new cells to grow, and it allows for these cells to either burst or die through the process of lysosomes, and it prevents these harmful cells from continuing to divide and multiply. By the lack of apoptosis, if we don't have apoptosis, this can lead to cancer, adverse events, etc. So we want to make sure that we have apoptosis to allow for new cells to come in. Okay, so now it's quiz time. Make sure to take a moment, read the following question, and let's try to answer this one together. Okay, now that you're done reading the question and you pause that video to ensure to come up with an answer, let's see if you got the correct answer. So the correct answer is C. It says if a cell does not pass G1 checkpoint, what happens? So remember, G1 has the G0 phase, which essentially is if G1 is not, that growth phase hasn't correctly grown, the cell hasn't grown properly, it will rest indefinitely before proceeding forward to the rest of the cell cycle. So that's what immediately happens. The S phase doesn't occur because we have that checkpoint there. If it doesn't pass, undergoes apoptosis immediately. Apoptosis can occur, but it's not a sure guarantee. You would go through the resting phase, and if the cell needs to go apoptosis, it will occur there, but it's not the immediate step. And then D is not great because it will skip to G2. Remember, we have G2, G0, then we also have the M phase in between. Then we have G2. So we forgot about the M, or not the M phase, sorry, the S phase, as they say, comes in between, right? So we have G1, S, and then G2. I said M instead of S. That was a mistake. But we have the S phase that essentially occurs in between, so it's not going to skip to G2. So that's why C is the best answer here. I hope this video helps, clears up some things. I know the CDK and cyclin can be a tad bit confusing because you have to see which one transitions where. Best to look at those graphs and the picture where you can see the transition between cyclin and CDK and the phases of the cell cycle. Make sure to check out our later videos or earlier videos by hitting that subscribe button, and also making sure to see our playlist for more details related to topics just like this. Hey, Geeking Out on STEM family, guess what? We want you to hit that subscribe button. Why? Because we'll be producing more content just like this for the AP Biology exam and the new digital SAT exam. By subscribing, you'll be getting exclusive, up-to-date tips, tricks, and secrets for both exams. Also, if you're a parent, a teacher, a homeschool instructor, or a student, you might be looking for affordable and exclusive digital products to help improve multiple choice questions, free response questions, or question bank questions related to both the digital SAT or AP Biology exam. We have them here. Make sure to check out our link tree in our bio for curated resources that you won't find anywhere else. These unique resources are designed to help you raise your score. By hitting the subscribe button, the more subscribers we get, by the way, the more videos we can make. So make sure to join our family to ensure that you get a perfect score for both exams.